So welcome everybody to episode 1 of Cell Lab. So you might not know what Cell Lab is, but I did play it earlier on my channel before I started editing. And hopefully it'll be way better when I edit it because, I sort of tinker around with things until, they work. But the basis of Cell Lab is that you get to use these different things and change the genome of a cell and try to do these various different tasks. This task right here is use the genome editor to make a species that thrives in environment of this challenge. When ready, put a cell on the substrate under the microscope and unfreeze it to see if it is visible. So this challenge is called algae, and we need to get our cells to a count of 100 to pass the challenge. There's a temperature thing that's freeze, observe, and incubate. Those are basically just like freeze, freeze is time, and then incubate, fast forwards time, observe is just regular. There's also a reset button. So in the genome, we see that the cell type is photocyte, which means that it like algae, it takes its energy from the sun. There's a bunch of different stuff there. You probably don't understand a lot of it, but yeah, you probably don't really need to. So for this, you get to place a certain amount of cells for different challenges. In this one, I think I get to place 10 cells. So mathematically speaking, each cell would be able to get 10, would have to get 10, or multiply itself by 10 to be able to complete the challenge. Oh, and there is a visual glitch, I didn't realize it at this point, where if you see that petri dish, that is not where it actually is. It's a lot to the left, which it looks like there's this invisible wall around the middle of the petri dish. So that's probably why I struggled, since you need to put them on the top to get the light, since they take light. But that's a problem because I thought that I couldn't put them in the middle, and that was just a thing that was about the petri dish and it visually glitching out. So if you don't know what adhesion is, it's basically a thing that is an option for when the cell splits. It sticks to or both of the children stick to each other and share nutrients. After tinkering around with it for like 10 minutes, I eventually, I think, looked at the hint and got them to place in the right spot, so I was starting to make progress. So after activating incubate, we successfully completed algae, the algae challenge. So here I save my genome for the cell, so I can use it later if I need it. So here I go to algae 2. So the 2 challenges and everything, and I did not go for the tutorial by the way. These challenges are sort of optional. This one is get your cells to 150. So, yeah, and this one, I'm pretty sure I literally just placed the same exact cell that I used last time, and it worked. So, yeah. So here I do algae 3, which is I get 5 cells to insert, and there's a lot less liquid or something in this substrate, and I have to get 120 cells. So if this one, it took me a while. So from what I'm looking at right now, I'm pretty sure I made a cell split into. So I had the first cell split into 2 other cells, which are both different, and then those cells create this I think softer cell that has less of a skeleton or something, or it's easier to produce, and so then I just have them produce it a bunch. Oh yeah, and so you can make so it's called M. So you can have M1 to M40, which you can make an M. So I made M1 have a child that is the first child be M3 and then the other one be M4. So that's how that works, and you can change them individually.